Hello everyone, I'm Flavia Bonalumi and I'm a PhD student at the University of Brighton, and I want to introduce you to my PhD work, which is titled The Development of a Multi-Layer Cryogel Bioreactor with Optimized Fluid Dynamics for Bioartificial Liver Application. Liver disease is a condition by which the liver is no longer able to fulfill its metabolic and synthetic function and is currently affecting almost 30 million people in Europe alone. There is unfortunately no other effective treatment other than transplantation. And for this reason, in the last decades, cellular approaches have been taken into study. In particular, extracorporeal devices are devices in which hepatocytes are seated in a bioreactor unit to provide the metabolic and detoxification function to the blood circulation. Few designs have been studied so far, but currently none of them have proved their efficacy in clinical trials. This is because to design an effective bioreactor unit for artificial liver is very challenging because of the choice of the biomaterials and cells, the high mass transfer required between cells and blood circulation, and the analysis of the fluid dynamics inside the scaffold to don't harm the cells in the bioreactor unit. So the first part of my PhD, of course, focused on the thinking of how to produce an innovative artificial liver unit. I chose cryogels as biomaterials to fill the bioreactor unit because these are supermicroporous hydrogels with an interconnected network of pores to provide liquid perfusion. I then decided to functionalize the surface of these cryogels with alginate, which is a negatively charged polysaccharide, which creates a very thick layer of water onto the cryogel surface, preventing the absorption of protein from the biological fluids. And this is very helpful to prevent pore occlusions and the failure of the device. Also, an RGD peptide was designed and inserted in the surface to provide anchorage for the hepatocytes to grow onto the cryogel surface. This is the range of characterization that I performed in the first part of my PhD. In particular, I was able to obtain cryogels with the pore size up to 100 micrometer required to host hepatocytes and to also support flow perfusion, with overall porosity and interconnectivity of pore of almost 90%. Protein absorption study proved that alginate modified cryogels prevented the absorption of protein from the surrounding biological fluids. And also here, you can see that the alginate modified cryogels were biocompatible, and the cells seated onto these modified cryogels were forming these large aggregates. This is not a situation that is wanted for this application because very large aggregates can prevent flow perfusion and occlude pores. In the RGD functionalized cryogels, as you can see, the hepatocytes spread onto the surface, and this is more wanted for this application because the porosity is still open and allow the perfusion of flow. After this, I set up a macro particle image velocimetry instrument, which was able to record the fluid passing through the cryogel pores. And as you can see here, these are glass beads at the same size of blood cells that flow inside the cryogel's porosity. From this video, I was able to track these beads flowing inside the pores, and with video post-processing, I was able to obtain the local velocities and vorticity sensed by the cells seated on the cryogel walls. And this was very useful to have a scale length of the shear stress sensed by cells and if that was harmful or not. Thank you very much for your attention, and bye-bye.